So it's now the time for the second talk. Is the speaker here?
Here.
Yes, okay. Nice to see you. Thank you very
Nice
much.
to see
So
you.
we come to the second talk of this afternoon, and the talk will be given by Jin Wang. He is from the Department of Chemistry, Stony Brook University in the United States. Professor Wang got his bachelor in physics at Yilin University in 1984. Then his physics degree in astrophysics at University of Illinois at Urbana University in Champaign. And in 1991, he became a postdoctoral at the and research fellow in chemistry and biophysics. 1996, he became a guest scientist at the National Institute of Health for 1 year. He became vice president and senior analyst at the Global Strategic Analytic Unit of the Citibank. Surprisingly, well, it's in economics in 1997 to 2004. He then joined to the State University of New York at Stony Brook as assistant professor, associate professor, and professor from 2004 to present. He was elected as a foreign fellow of European Academy of Science and a fellow of the American Association of Advancements in Science, a fellow of the American Physical Society. His main scientific contributions, which are very large, cover a large area using methods, in fact, of statistical physics and molecular simulations applied, and also here in his talk, to biological systems as complex systems. Today, so we see we come from a social problem to a biological problem by considering the living organic system and to understand how such a system can be described by physical concepts. So the title of his talk is Non-Equilibrium Physics in Biology. Please, we are very interested. Okay, thank you.
Thank you very much for Professor Falk's introduction. I would also like to thank Yuri for the kind invitation, and my heart goes to the people in the war, and I hope the war will be finished soon and the peace will recover. So let me share my screen.
I see now landscape and flux. So
okay, good.
Now you make full screen.
Yes. So now it's full screen now. Okay.
Okay.
Great. Finally, it worked. I tried a few times. Okay. Thank you very much. And here's the title of my talk, The Landscape and Flux of the Biological System. So this is our approach that we started like about 15 years ago. So I'm going to, let's see. I'm going to give an overview of this framework, essentially using the non-equilibrium statistical physics to look at the global dynamics and thermodynamics. So I'm going to talk about some theoretical aspects, and then I'm going to talk about a few biological applications. And at the end, I'm going to talk about some of the experimental tests of these ideas. So here's a illustration graph that you often see in the biological textbook about a cell. This is the smallest unit of living objects, and even with this cell, we know that this is not an equilibrium system because it constantly receives information, material, and energy from outside or the environment, and to perform certain functions. So, for example, signal transduction, going from the surface of the cell, receiving the. So, as I said, this is the smallest living unit, and by receiving energy input and material input, it performs the biological function of signal transduction, then eventually then reach to the genes so that you can make some changes or modifications of the genes so that can have the response of the genes towards the outside perturbation. And this is a living cell in response to the outside perturbations that Carolyn talked about in terms of other systems. But this within the cell, there are not just one molecules, but many molecules. For example, there are many proteins, mRNAs, and etc. So one can crudely think about this system, a small living cell, as a compartment which has many chemical reactions, because those molecules interact with each other. So you can think about it as a chemical reaction soup. So many different kinds of molecules interact with each other, form a chemical reaction network, and it's a chemical reaction soup. So in this sense, one can, in the physicist language, write down the equations of these chemical reactions. And of course, this is very complicated chemical reactions. The X represents, say, for example, the concentration of a specific species of the molecules, like for example, proteins or ligands. On the right-hand side is the chemical reactions, a reaction driving force. And you can think about this as a high-dimensional objects with different species, concentrations of different species of proteins and molecules. And on the right-hand side is the interactions among those molecules, essentially the chemical reactions inside the cell. And typically, how you analyze these, you realize this is a nonlinear dynamics 101. This is the typical nonlinear dynamic equations. And the way to do it is you find the fixed point, x dot equals 0, and do the linear stability analysis around those fixed points. And the outcome is that you can find out the local stability analysis around those fixed points. But those information are local, means that you can only know the information around those fixed points locally. However, for many cases that we are interested in, not only for the local information around those fixed points, but we would like to go from one fixed point to the other. For example, Carolyn talked about it, like stem cell differentiation, going from stem cell to differentiate cell, from one basin to another, one fixed point to the other. In this sense, you want to know the global information, not just the local information. You want to get out from the local information. So this global stability information is missing from this local stability analysis, the typical one. But it is essential for characterizing the whole system. So, and can we find a global description of these complex systems? Well, let me give an example of how we treat or view this problem. So suppose you have, like, this could be genes or proteins or some molecule species and a high dimensional space, as we can see, I mean, represent the expression or concentrations of the proteins. Then for this dynamical system, any state in the high dimensional state space represents a particular combination of this variable or the concentrations. And then you go from one state to the other in this high dimensional state space. This represents the evolution of the system from one state to the other. And if you have a gene, a genome, and DNA, which has lots of genes, and all these genes are interacting with each other, form a gene network inside the cell. And then this network, for example, each of these particular gene or protein or expressions will have certain values So any of this kind of combination of different values give you one particular state. And another combination of these high and low expressions will locate into another state. So the question is, how do you know that at one moment you are at this high dimensional state space, you go from here to the next moment to here? Why do they go from here to here? Of course, if you can monitor the high dimensional dynamics, you would like to know not only know that it goes here, but also why it goes here. Of course, if you have an underlying landscape, then this is not hard to understand. If you have a global landscape, so for example, this is representing this state, this is representing that state, it's not very hard for you to understand why this state you go from the top to the bottom, because the landscape drives you down to the gradient globally, and locally there is a gradient force driving you down there. So this is, if you have a landscape like this, you can globally characterize the whole system's dynamics rather than just look at the trajectory and wonder why they go from one place to the other. Now, then the next question is, does this landscape exist? Well, in the equilibrium physics, this is relatively straightforward, because if you know the interaction energy, we know the Boltzmann law, the equilibrium probability is exponentially related to this landscape or the interactions. And what that says is, since the probability or equilibrium probability is related to this potential, once you know the interaction, you know the weight of the state at each location of the state. What that means is that if you have two fixed points, how do you connect one to the other? Because if you know the all the possible states in between and know the weight in between, then you can connect them together, and then that can determine the stability and how you go from one to the other, because you have the connectivity of these different weights of the states together. On the other hand, the gradient of the interaction gives you the next moment where you are going to go. So these two laws of equilibrium statistical physics governs the global stability and also the local dynamics. Now, there are a lot of applications of these ideas to the biology, such as the evolution, differentiation, development, protein dynamics, folding, binding, etc. All of this assumes that we know the energy function a priori, and the gradient of which is the driving force for the dynamics. However, if we look at the cellular network, like gene regulatory network or chemical reaction networks within the cell, it's not very hard to find out that the driving force in general is not a pure gradient of a potential. So one can check these chemical reaction equations within the cell, and it's typically not a pure gradient of a potential. So this is the sort of the fundamental issue related to how to globally characterizing the complex systems such as the gene regulatory networks within the cell. Of course, then one can borrow some of the non-equilibrium physics ideas of these pioneers, the Prigogine, Lebowitz, Graham, Hagen, Hangi, Zier, all these people that I can't list them all, and find out how we sort of look at this problem. So let me go directly to the issue without talking too much more on the other people's work, but just concentrate on our approach. What we have found in back about 15 years ago is the following. although you cannot write down this driving force, this is the general equation. You cannot write down the driving force as a pure gradient, but you can. I mean, what we have seen, we have shown is you can write that as a gradient force plus something else. And you may wonder, of course, you can write down this way. You can write down as a gradient plus something else. But this something else is not some arbitrary force. It turns out this one has very specific meaning. First of all, it represents or it's directly related to the net input of either energy, material, or information to the system. In other words, this represents the net input to the system. In other words, this represents whether or not the system is in equilibrium or not. Because in equilibrium system, there's no net input or output. But once you have a net input or output not equal to zero, this represents the detail balance breaking, means that you are not at the equilibrium. So that's the first thing. The second thing is this force is actually rotational, which is very interesting too. So this part is, so the force, the driving force, can always be decomposed into two components. One is the gradient, the other is more rotational-like. I'll skip some of the details of the mathematical derivation, but just saying that so the equilibrium dynamics looks like a multi-dimensional state space in analogy to a charged particle moving in the electric field in the multi-dimensional state space. But the non-equilibrium system, this is a closed system, but the non-equilibrium system, the open system with the exchange of net energy or material information, then the steady state probability defines you the landscape. But the driving force is not just determined by the steady state probability, but it also is determined by a rotational flux force. So it looks like in the multi-dimensional state space, the open system with net input behave like a charged particle moving in an electric field and magnetic field. So there's a spiral downhill trajectories. So in other words, so this is the sort of the dynamics analogy. On the other hand, one can write down a zero noise expansion and show that the gradient part of the landscape indeed is a so-called Lyapunov function, which can be used to characterize the global stability. This is the gradient part. On the other hand, what is the sort of the importance of the other parts, which is the rotational component part of the driving force? Here's the illustration. Usually you will say, if you have a landscape and you go from one state to the other through a saddle point, so going forward and backward, you all follow this minimum energy path. And the rate is determined by the barrier height in between these 2 points. However, once you have the flux or this rotational component of the driving force, you're not going to go through typically in the finite noise limit to the transition state. Instead, you go around. And furthermore, going backwards, you don't follow the same route, you going through a different route. So since they don't pass the transition state, you can't use this as a marker for determining the transition rate going from one state to the other. You have to determine the optimal transition rate along the path, along the specific path. So that's the lesson we learned. The other lesson we learned is the typical fluctuation dissipation theorem is not going to work. In the equilibrium, we know that if you kick the system out of the equilibrium, watch the response, that is going to be equal to the equilibrium fluctuations. However, once you have the net input or output for an open system, then if you kick the system out of the steady state, then the response is not just going to be the the steady state fluctuation. It will also have a component contributing from this rotational component of the driving force. Also, the thermodynamics is going to be changed because the total entropy production rate is not going to be just equal to the negative of the free energy change for the equilibrium system with zero net input. It will have additional contribution, so-called housekeeping. This is really to keep the steady state. To keep the steady state, you need to have the energy input represented by the net flux that's supporting this this steady state. There's a cost, thermodynamic cost associated with that. So this is all a little bit arbitrary, abstract, but let me give you an example and showing that this is in action. This is a mammalian cell cycle. It's basically all this component is crucial. These molecules cycling and CDK complex at different type, 1, 2, 3 different like D, E, A, and B. They at different types they control. I mean, they interact with the other molecular species, and they control the progression of the cell cycle. Why we are so interested in the cell cycle? Because as the smallest living objects of the living cell, the living cell has, according to the definition of living, we at least they should have some properties, which is to copy itself, sort of to make the next generation. I mean, this is a difference between living and non-living. At least one aspect that differentiates living and non-living is to copy yourself or make it to the next generation. So this is an essential function for the living objects. And this living, the smallest unit of living object, the living cell, has this feature that copies itself to the next generation, so that it can expand its population. And this is tightly controlled by the underlying interactions of the molecular species of the cycling CDK and among with the others. So this is a molecular network that interacting with each other. And so the cell cycle experiences several stages: G1, SG2, and M phase, and completes the cycle. So here's the biological textbook of illustrating the cycle. The typical cell cycle experiencing a few phases: G0, G1, through checkpoint SG2 and M. So essentially, completes the cycle. They at the end of the cell cycle, they divide the cell into two. So then you can keep on going this cycle again, 1 divided by 2, 2 divided to 4, etc., etc., so that you can copy the information down to the next generation. So this is the real cycle going around. So in terms of physical process, here's we want to describe that globally. Here's the vertical one, is the minus log of the steady state probability distribution. So in other words, this is the landscape, I would say. These two axes represent the gene expression or the protein concentrations of the two selected ones. So what you can see, this is the landscape of the shape of this cell cycle, is actually an irregular Mexican head. Mexican head has the shape of the center is high and the other outskirt is high, but in the middle there's a closed ring valley which is relatively low, corresponding to the low potential. And if you stay at anywhere along this part of the state space, because the gradient of the landscape, you get attracted along this ring valley. But if the driving force of the whole process is only determined by the gradient of this driving force, you are going to sit down at the G0, G1 state and stay there forever. However, we know that the cell cycle goes around and around and around, and so this completes the cycle. So there's another driving force, the other component of driving force, which is the rotational driving force, which is represented by the purple line, to drives that around to form a coherent oscillations. So this is our picture of showing how a limit cycle can come about, emerge, and how these two forces act together to form a coherent oscillations, to form a fundamental function of the living cell, which is copying itself and and passing it to the next generation. So you may wonder why this is important. The important aspect is the following. Since the landscape is global, so I can use the global information of this landscape to ask the question, what is the underneath structure that can change this landscape global shape significantly, so that can influence the function. For example, this cell cycle goes around, even along this closed ring valley, it doesn't go through this sort of uniformly, because there's a valley here corresponding to the G0, G1. This valley here corresponding to SG2 phase, and this valley here corresponding to the M phase. And in between, you have this local barrier, this local barrier, for example, is, although I call it a local barrier, it's actually a global on the landscape. Will determine its will influence the speed of the cell cycle. Also, the quality of the cell cycle is determined by the height of the center island, because if the center island height is low, then you can go from here to here very easily, and then you lose the sense of direction. You don't know where it comes from here or from here, and so this is also an important measure of the quality of the oscillation, so you can have a coherent oscillation. So this is so as I mentioned, that all these are important because the global flux is also important to drive that, to make sure it goes around to complete the oscillation. So all these are important to determine the quality of the oscillation and the speed of the oscillation. Why the speed of oscillation, the quality of oscillation is so important? First of all, it's important to make sure that you can perform the biological function of copying itself. Second of all, the speed of the cell cycle is a hallmark of cancer. As we know that cancer cells has a much higher speed of splitting or division than the normal cell. So one could ask the question, when you will change from a normal state of this landscape to a more flatter landscape of the cancer, and which component of the underlying network will contribute to this. So one can use, for example, the height of the barrier and the magnitude of flux and the local height of the barrier to understand and explore the underlying structures of the network. And in fact, we found that even for this network, we can find that these blue ones are the V's, and CDC24PRB are the key genes, and this line represents the key regulations. Arrow represents the activation, and the bar represents the repression. All these interactions with the color and the genes with the color are the key ones. We can identify those are the key ones. Those are the targets for possible targets for the cancer, for example, or influencing the cell cycle speed and the quality. Okay, so that's why it's useful. for this. This is purely dynamic descriptions. How much time do I have?
You have, oh my God, for 20 minutes.
20 more minutes. Okay,
Yes.
great. So another example is what Caroline mentioned, is the differentiation and development. So I will briefly mention about this, because we talk about the example of the cell cycle or limit cycle oscillations, but the other possibility is multi-stable state. In 1950s, Waddington asked his friend to draw his artistic friend to draw a picture of his idea. He thinks that the differentiation and development starts with a ball, like a marble, on a hilly landscape, and the differentiation process is basically look like the marble rolling down to the hill and going to the different branches or different local valleys that forms the different differentiated states. So that's his sort of picture of a so-called Waddington picture of the epigenetics or differentiation development, which influenced a lot of thinking of the field. But our question is to ask whether or not this can be quantified. So we took a critical circuit that often appeared again and again in the differentiation development, which is this is a gene module or gene circuit module, which is two genes mutually repressing each other and self-activating each other. And so what happened is that this circuit, although this is this circuit module looks relatively simple, but it has all the essential features of the Waddington landscape. Here's what we are showing. This X represents a composite axis of the X1 and X2. You can think about this as sort of like a combination of X1 and X2. Since we only have 3 dimensions to represent, we can only use one for the gene expression. And the other one is the strength of the self-activation. And we know that during this differentiation and development, this strength of the interaction actually changes along the way. So you can think about at the very beginning, the cell is sitting at the stem cell state. I mean, it's the basin of traction. But during the developmental process, since the self-activation strength is changing, so the landscape shape keeps on changing along this direction. So eventually, at the end, the differentiation process proceeds. Eventually, the landscape changes from one basin of traction to the 2 basins of traction, and form the differentiated states at the end. And the forward differentiation and backward reprogramming paths are different. So this is sort of things that we now finally can represent both the dynamical. This is a dynamical landscape of the differentiation development, and also the path of differentiation and reprogramming. They are different because of this rotational driving force component that we mentioned. So even at the multi-stable states, both the landscape and the rotational component of the driving force are important. And then we look at a more sort of going beyond the circuit module. We look at more involved network, which has the stem cell markers and differentiating cell markers and all the other that form a gene regulatory network. And then we study this network features. And we can find that you have a sort of stem cell state, and you can have a cluster of differentiated cell state. And going forward, these the differentiation process and the reprogramming process are quite different. And the remarkable thing is we can do the global sensitivity analysis on this landscape, and in the past we can identify the key genes and key regulations responsible for the differentiation and reprogramming process. So this octal shape and the solid lines representing the key genes or the key regulations responsible for differentiation and reprogramming. And remarkably, one of these, I mean, some of them are consistent with what people have already found, but one of them is something that people haven't been realized, is so-called PBX1. This turns out that the interactions are important. The gene itself is important because it interacts with another key component of the differentiation reprogramming, which is a non-org. And PBX1 gene is not very well known for participating in this process, but we identify that as an important component. And an experimental group took it seriously, and what they have found, they did a series of experiments, and what they have found is including the PBX1 can enhance the reprogramming, the backward of the differentiation process. That's number one feature of the PBX1. The other one is they found that the PBX1 is a therapeutic target for the melanoma, which is also very important for a marker. And the next, what they have found is that it has the function, the PBX1 has the function of rescued hair follicle mesenchymal stem cells synesis and apoptosis. So you can sort of make, as we call it, make things going back to the young stage, where it makes things younger. So, and this group of experimentalists are now trying to establish a company based on that. So this is a very exciting prediction that we have from this analysis on. And since my time is limited, I just want to mention this idea can be used on the cancer networks and look at the cancer landscapes and find out that the key genes and the regulations for the cancer. Another application is this could also be used to study the aging process, and we can think about aging as some of the combination of many different factors. We know some of the people knows that there could be a nutrition reasonings and telomere reasonings and genome instability, epigenetic alteration. But if you consider try to consider them all together into a network, one could also form a relatively a simple network, I mean simplified network, and try to study the effect of this network and the underlying landscape of the aging process. And we can also quantify the landscape of aging, so you can find sometimes you could have the rejuvenation stage and also the aging state. It's not necessarily always the aging state, but one could in principle have an emergence of the rejuvenation state. And the interesting thing that we have found in these regulations, we could also find the rejuvenation state and aging state can actually oscillating around each other. So sometimes they can rejuvenate, sometimes they can be aging, so they can go around. And of course, there's a lot of thermodynamic costs associated with that. And we thought this might be crazy, because this indicates that one could go around and oscillate between the old and the young. And this seems to be odd. I mean, we never see something like this. But oddly enough, we found one example that this is a kind of jellyfish, but it only lives in the South Japanese Sea, and it has the shape like this. And this is a very interesting, specific type of jellyfish, and this creature seems to be very interesting, because it seems that this creature is the only creature in the world that looks like it grow from the young stage to the old, and then once it's reached old, it goes back to the young stage, and then going back to the old stage and going back to the young stage. This is the only creature that people are arguing about whether or not this is called immortality, because this special jellyfish looks like having this oscillatory behavior of between young and old, and young and old keep on going. So we found this one example, and maybe the model that we have is not totally nonsense. So this is something that we are currently very interested in looking at it further. And one of the last aspect that I want to mention is that for the evolution, because we all know that Darwin says the fittest survive, and the quantitative description of this was given by Seward Wright and Fisher, and in terms of Wright, it's called a fitness landscape. Wright's fitness landscape means that the evolution is always going towards the optimal, and this is the fitness optimal, or the fitness peak. And in terms of physics language, you can invert that back to the energy, and so they always go down. I mean, in terms of energy, always go up. In terms of fitness, is quite easy to understand. And for Fisher's fundamental theorem of natural selection is that the adaptation rate is always going up. So it's representing similar ideas. And these ideas has been quantified, as I said, as the quantification of the Darwin ideas, but it has some criticism also. And this criticism was coming from this guy called Van Valen. He used the Alice in Wonderland as the example of showing this so-called Red-Queen hypothesis. Means in the Alice in Wonderland, there's always old saying that in order to survive, you have to run all the times. So in other words, this can be better illustrated as the Escher staircase. You can go around, it looks like you never end, and it's just keep on going at the end. In biology, you can also have the situation like that. If you have a lot of interaction between species, such as co-evolution, like a wolf and sheep, the sort of co-evolution, if you have this co-evolution process going on, you can run into the cycles. Once you have these cycles, how we are going to say that the evolution always goes to the optimal, because there's no, it looks like you can go back to yourself and which one is the optimal. And when you reach this cycle, is the evolution already stopped and finished? So these are all the issues that related to this Darwinism, the fit and survive, coming from the Wright and Fisher's ideas. So we can illustrate this as one aspect of the Wright and Fisher's issue, because we realize that the Wright and Fisher's this mathematical statement is based on highly simplified scenarios. So for very, very simplified scenario, you can have a situation like a gradient landscape where you always climb up or going down on the energy. However, if you have any kind of complexity in it, such as the coevolution in it, this would not hold. And the reason is also because this rotational driving force coming in and play an important role. And in terms of Fisher's fundamental theorem of selection, basically this is the sort of adaptation. The adaptation rate, the fundamental theorem of selection says is equal to the genetic variance. And so if the evolution reach the end or reach the optimal point, the genetic variance is zero, so there's no evolution. However, as we just mentioned, the coevolution, even if you reach the optimal, they may not stop the evolution. This is because there is another contribution coming from this rotational component of the driving force, so that even if you reach the optimal, so for example, you reach the cycle, this can be zero, but the genetic variance still there because you have this rotational driving force. And so this is our interpretation, where you can reach the optimal or sort of the cycle of the evolution, reach the optimal, but the evolution still goes on because you have this rotational driving force push you around, so you still have genetic variances. The evolution still keeps on going even if you reach the optimal. So we have applied this idea to many systems, going from intracellular network to intercellular network to evolution, and even to ecology, game theory, and the chaos, and even non-living system turbulence and non-equilibrium phase transition, which I don't have time to talk about, and some of the quantum dynamics and space-time dynamics. But let me, in the remaining few minutes, let me just show you this. Right now, we just talked about theory, but there are quite a few experimental studies that showing the quantification of these two components of driving force. I'll just give you two examples. One is the simplest possible gene regulatory circuit that people are interested in. It's called the lambda phage circuit, and which is involve two genes mutually repressing each other, and one can sort of expect that you could have like two states. One is C1 dominate, the other is CRO dominates. And by applying some of the molecular biology techniques, one can label these two genes differently according to their locations. Essentially, one sort of genes are related to, one sort of the genes are labeled and attached to the cell surface. The other genes is labeled and is sitting in the middle of the cell, so that they can distinguish these two genes in one cell based on the location, so they can sort of separate these locations. So in this way, they can, by looking at the fluorescence light, they can look at in time these states, different states. So this different color representing different states. So for example, the purple ones representing both C1 and CRO are enriched, and the red means that both are not present, very small, and the blue is the CRO is more, and the C1 is almost none, and the blue, that's the blue, and there's also a green one, which is the C1 is more, and the blue and the CRO is almost none. So you can actually watch in time, and you see this four states looks like four states switching from one to the other pretty frequently. And if you look at the histogram, this is best showing that in addition to what you expect, the two states, right? The histogram of the fluorescent signals, you have two states. but you also have another two additional states. One is 0, 0, the other is 1, 1 sort of state. And interestingly enough, this system, I mean, used to be, people call that hydrogen atom of the molecular biology because it's simple, because they think that this mutually repressing state has only two states, 1, 0, 0, 1 state. But in fact, what the experimental is showing is not entirely, even for this simplest hydrogen atom, it's not that simple, because it could have 4 possible states with the temperature mutant. And because it's very stable, so they use the temperature mutant. And interestingly enough, they don't go directly from one state to the other. They actually go over the bridge from one to the other. They don't go. This is not a good way of going from one state to the other. And they going from one intermediate state to the other. It seems to be the best way of going from one state to the other. So this is quite surprising from the point of view of the simple system can have some complex behavior. But nevertheless, one can quantify their landscape. It's actually 4 basins of fractions. Anyway, the last example I want to show is that that this is about the landscape quantification in the experiment. But how about, one could ask the question, how about the rotational component? And the rotational component, we can ask the question, how do we quantify that? And for a molecular reaction called an enzymatic reaction, we know that 2 molecules can meet, and then the enzyme can catalyze the substrate and release the product. And there's an empirical law called the Michaelis-Menten law. It's the rate of this catalysis, inverse of this rate is inversely related to the substrate concentrations. And the experimental collaborators figure out a system which is very interesting. This is the horseradish peroxide is the enzyme that catalyzes 2 substrate to 2 products. And horseradish peroxide has conformation changes. So in fact, in terms of physics language, there are 2 states of this enzyme. Through the catalysis can catalyze one substrate to the product state and catalyze another substrate and back to the end. Long story too short, one can show that this is a relatively simple description that shows that sometimes at certain conditions that the internal flow, this flux, the rotational component of the driving force, if that's equal to 0, then we go back to the Michaelis-Menten rate, the normal rate. However, if this internal flow or the rotational component, the driving force, is not equal to 0, I mean, there's a net flow going around this component, then you are going to have an additional term that depends on the substrate concentration. And these 2 behaviors are very different. So they did a single molecule enzymatic reaction test on this, and this is the enzyme, and this is the essentially the substrate molecule, and this is the product. And once you form the product, the fluorescence flash out, so you can see the signals. And they do the cross correlations, and then the important aspect is that what they have found is that the inverse of the catalysis rate versus the sort of the substrate concentration, if the inverse of the substrate concentration, if it's Michaelis-Menten traditional rate, it should be straight line. But what we have seen is not a straight line at all. All these curves are very much deviating from the straight line, indicating that this rotational component is not equal to 0. And then from there, you can infer what this rotational flux or rotational force is, and it actually depends on the concentrations. It's not zero. Not only that, from that you can also infer what is the chemical potential of the driving force for this reaction. Essentially, they are sitting at some 9 kBt, and also from there you can calculate from the experimental data and find out what the entropy production rate is. And this is the dissipation cost that supporting this enzymatic reaction, which has a non-Michaelis-Menten behavior. And finally, the time irreversibility is associated with the flux. So the larger the flux, the time irreversibility.
We should allow some time for discussion. So
I'm just finished. Yeah. So as you can see, we can in this example we can use these the experiment to quantify the flux, the rotational component of the driving force. And here's a comparison between the equilibrium and non-equilibrium system and different aspects of that. And finally, let me thank my collaborators. There are quite a few of them over the years in establishing the landscape and flux approaches. And also we discuss about how we use the lambda phage to quantify the landscape of the cell fate, and using single molecule for characterizing the non-Michaelis-Menten rate to quantify the flux. And thanks my support, and thank you for your attention.
Okay, and thank you very much for this very interesting talk, and also very relevant talk for understanding living systems. And please wave your hand if you have some discussion contribution. Yes, Caroline, please. Caroline?
Yes, I think, yes, I'm here. I think you can hear me. Yes. Thank you very much, Jin, for your very inspiring talk. And now I understand more the questions you were asking earlier on. Can I just expose my ignorance? So there was one thing I wanted to ask you about local versus global stability. Yeah. Am I correct in thinking that global stability is the sum of the local stabilities, or is there something more to it?
Not quite. So let's go very much back to this because okay, local stability. I actually mean that you do x dot equal to 0 and find the fixed point, and then around the fixed point you do the sort of the linear sort of a perturbation, and so you can basically judge whether or not it's stable locally or not. By global, I mean that whether or not you can find for example, a Lyapunov function for global stability that you can always go down on the stream. So when I say global stability, I say how it's more or less more like how you connect from one state to the other, because this is a local stability analysis around basin. This is another local stability around basin. But how you connect from one to the other, the local stability analysis wouldn't give it to you. This barrier separating these two cannot be obtained from just the local stability analysis around these fixed points.
Okay, that's great. That makes sense.
Yeah.
And then
you
need
you need the barrier separating them. This information is missing in the local stability analysis.
Makes perfect sense. Okay, great. And can you say, on this particular slide, a few more words how you get from the network representation of your system to a landscape? I mean, you say there isn't actually, it's not representable as a gradient. You have this additional term, but as a first step,
So
how would you even do that?
Okay. Let me. I skip a lot of things, but here's the thing. Essentially, I mean, if you give an example, if you have X is abstract concentration space, you have Fx. So basically, how you get the landscape, you put some noise on it, and this is usually the case. You always have some noise, non-zero noise, and this becomes a stochastic equation, and you can't rely on this x because x is stochastic now. But what you can reliably rely on is the probability or patterns of the trajectories, because the patterns of the trajectory satisfy a deterministic equation, and it's linear. It's linear in P. Yeah. So the patterns or the probability is predictable and it's linear, and that's why it's predictable. These guys, even without the noise, is maybe not predictable because of the nonlinear dynamics. But with the noise, it's even more unpredictable. But the patterns are predictable. And this pattern, the probability distribution at the steady state, if we get it, that's the thing I call the landscape. And the minus log of the steady state probability, I call that landscape. Of course, in the equilibrium, the minus log, steady state probability distribution in the equilibrium case with zero net flux becomes equilibrium probability. So that's the analogy here. But in the in general, if the net flux is not equal to zero, the steady state probability distribution, the steady state doesn't mean equilibrium state. But the landscape definition is general because I just take the minus log of the steady state probability distribution as the landscape.
Right.
Either works at equilibrium or works in the non-equilibrium case.
Great, very interesting. Thank you very much.
Yeah. Okay, please, Yuri.
Yeah, also I add my thanks. Thank you very much. And the question is not directly about what you were speaking, but you know, when you started to speak about fluxes, flows, linear stability analysis, attraction points, I cannot stop putting the question, do you see some universality in these attraction points? Could you speak about some categories of attraction points you have in your flows? As you know, we have fixed points in randomization group analysis of critical behavior.
Yeah. This
so to say, different systems being governed by the same Lyapunov exponents in your linear stability analysis.
Yeah, this is a very interesting question. This is still an ongoing project that we are trying to investigate. Because once you have the flux, then there's an additional component driving, which is rotational. And the question is, can you have the same sort of phase transitions that we have in the equilibrium system? And I think for some cases, you can still have this similar universal phase transition as the equilibrium phase transition, corresponding equilibrium phase transition. On the other hand, you could have some new phases that the equilibrium systems doesn't have. So, for example, limit cycle. The limit cycle cannot exist in the equilibrium. In other words, in the equilibrium system, there can never be a limit cycle phase. So this could be understood as a non-equilibrium phase or a phase transition going from one possible state to the limit cycle phase. So there could be non-equilibrium phase transition just for non-equilibrium systems, but there could also be some equilibrium counterpart that in the equilibrium system there is a phase transition, but in the non-equilibrium cases, this equilibrium phase transition is still there. And I suppose, although I'm not very sure that the critical components might be similar, but we have to explore more about this. But certainly there are some new phases and new critical components or new critical behavior coming from.
Yeah, thank you. May I put another question, very short? As far as I understand, in this dynamical equation, this very bright and crucial idea was you had gradient-like term, and then you have rotational-like term,
Yes.
and then you proceed. And then the question would be, is tertium non-datur, so to say? Could one think about the third term for some reasons, qualitatively different? Yeah.
Well, okay. To make things a little bit sophisticated, there could be a third term. This third term comes in if this term. Okay, for example, if the noise is this d. I mean, right now is for simplicity, I set the d as a constant. However, if the d is x dependent, which is very likely the case where when you have the sort of the intrinsic noise, that if you start with the intrinsic noise, this d in general depends on x. It's state dependent. If you have state dependent force, diffusion, then there's this force has another component that is associated with the derivative of this d with respect to x. So there's another component. There could be another component if the d is x dependent. Yeah.
Thank you.
Sure.
There was another question by Yuri Suchowski, please.
Please, yeah.
Suchowski?
You have to switch on the microphone.
Switch on the microphone, yes. Okay. If this is not the case, I have a short question to these surprising jellyfish oscillations.
Okay. Yeah, we are surprised too.
Yeah. So, do you have any idea how you can, I mean, do you know anything about the aging of these oscillatory systems? Is there any chemical indication you can see that how many oscillations have been made? I mean,
well, first of all, this system has been very limited studies on it, because I think it's because, first of all, somehow this specific jellyfish only lives in the South Japanese Sea and is pretty fragile. I mean, I think it's very easy to destroy them. But on the other hand, if they sit in a comfortable environment, they can sit for a long time. Many oscillations. In fact, if the people report that, saying that that's why they call that this question mark whether or not it's immortal, because people have seen in the comfortable environment they can live many cycles, and it's almost countless cycles. So that's why they say whether or not they are asking if it's immortal. Yeah.
Okay, thank you.
Yeah.
So
but I think it's a great idea for people to. I certainly would wish that more people are studying this system, because certainly this is the only one that so far is young growing to old and old going back to young. I mean, literally the size shrinks when going from old to young and back. So.
Okay, now Yuri Suchowski, you can ask your question. You have to turn on your microphone.
Switch on the microphone. Yeah.
Switch on the microphone. Yuri Suchowski. No, it seems not to be possible.
To switch on, yeah.
Are there other questions at the moment? If not, I would like to thank again for this really interesting talk, and I hope I see you then tomorrow in the next session. Okay, Yuri, you want to say something?
Just to thank the speakers and the chairperson.
Yes.
Okay.
I think Oksana has an announcement. Do you have, Oksana?
Yes, I have an announcement. We need to make a group photo for this Ising lectures, and we decided to do this on every day. But there is a limitation in Jitsi for switched on cameras, only 10. So therefore, we made a separate meeting in Zoom where we can meet now. I sent an invitation and link in chat. So we welcome everyone to switch to this meeting in order to make a group photo. Thank you.
Okay, pleasure.
So I'm
So-
already waiting you there. So
Yeah, I go to Zoom. See you soon.
Yes,
see you
I go
soon.
to Zoom too.
And goodbye from Jitsi.
Bye. Bye, everybody. Thank you very much.
Thank you.
Bye bye. Thank you very much.
Thank you.